the title, phloem and translocation. First of all, I would like to ask, do you know what is the phloem? And do you know how it works? Let us learn it. Actually, phloem is composed of still living cells that transport of organic nutrients, particularly sucrose. In vascular plants, phloem carries sucrose to all parts of the plant or into storage structures. The phloem is concerned mainly with the transport of soluble organic material made during photosynthesis, which called translocation. I would like to ask you, do you know how the phloem looks like? And do you know the structure of the phloem? The structure of phloem tissue consists of sieve elements and also component cells. Conducting cells is also known as sieve elements. Let us know the details about the conducting cell. Sieve elements are responsible for moving sugar ladder fluids through the plant. Sieve elements are arranged end to end to form cylindrical sieve tubes. The sieve elements are separated by sieve plates. These perforations allow the cytoplasm to flow from one sieve cell to the next in the form of cytoplasmic strands. Now, let us learn about component cell. Component cells is also known as transfer cell. The survival of sieve tube members depends on a close association with the component cells. The cytoplasm of a component cell is connected to the sieve tube element by plasmodesmata. It contains nucleus and has a larger number of ribosomes and also mitochondria. Now, I would like to ask, do you know what is translocation? If you don't know, it's okay. I will show you now. Translocation is regarded as an active process. The energy required comes from component cells. Generally, the sucrose will enter the phloem sieve tubes by active transport. From sieve tubes, the solids are transported to the other parts of the plant that consume or convert it into starch for storage. There are many hypotheses have been proposed to explain the movement of the products of photosynthesis through the phloem sieve tubes. One of them is mass pressure flow or mass flow hypothesis. The mass pressure or mass flow hypothesis is proposed by Ernst Mach in 1930. According to the hypothesis, the sucrose will move passively in the phloem sieve tubes due to the difference in water potential between the leaves and the roots. The high concentration of sugar in the sieve tube created low or more negative water potential in the leaves. Thus, the water will enter the phloem from the xylem and create a high hydrostatic pressure. The concentration of free sugar in sugar sink is always lower than that found in the phloem. Sugar sink is the part of plant that consumes or stores sugar. The concentration gradient allows the sugar to diffuse out of phloem into sink tissue. Then, a low water potential is created outside the sieve tubes. The hydrostatic pressure gradient formed between the source and sink drives the mass flow of water and dissolved solids. The source is referred to the leaf cells, where the sink is referred to the root cells. If sugar continues to produce and converted to starch or oxidize, the gradient maintained and the mass flow will continue. In plants, the continuous flow of fluid is maintained by the secretion of sugar and the outflow of the fluid. Here are some evidence to support the pressure flow hypothesis. A positive concentration gradient between the leaves and the root of various types of plants has been detected by the scientists. The concentration gradient disappears if the leaves are removed. When the leaves are first removed, the fluid from the phloem flows out fast at the beginning, but gradually slows down. However, there are also some evidences to against the pressure flow hypothesis. There is possibility of a two-way translocation in the phloem. However, the mass pressure hypothesis is advocate only a one-way flow, whereas solids move in various directions. And this leads to the assumption that the solids move in direction of the shoot and the direction of the root. The sieve plate could have been lost if it was exhibiting any particular function or process of the plants. There is evidence to show the translocation is influenced by the temperature and also metabolic poisons. This implies that the translocation of sugar, amino acids, and other solids is an active process. Okay, now do you understand about the phloem and translocation? If you still don't understand, here are a video clip which can help you to understand it.